Welcome to the deep dive, where we drill down into complex biological research to find those moments that, fundamentally change how we understand the body.
Easier.
Today, we are talking about energy. Specifically, an energy crisis that faces your longest, hardest-working nerve cells and the really surprising way they survive their incredibly demanding jobs.
We're diving into a fascinating intersection of cellular metabolism and some really debilitating chronic pain conditions.
Right.
Our focus is on the primary sensory neurons in your dorsal root ganglia, or DRG. And to really get the scale of their challenge, just think about this. These neurons transmit all your sensory inputs, pain, touch, temperature, through axons that can stretch nearly a meter long in an adult.
A meter-long cellular cable, that is. It's just an unbelievable distance for a single cell to maintain.
It's a massive energy requirement.
Just to keep the lights on, right? Let alone handle all the constant electrical signaling.
It's a constant marathon. Every single action potential, every signal, it demands a huge continuous supply of energy. And that energy comes from mitochondria, the cell's powerhouses.
And when that supply chain breaks down,
that's when things go wrong. The distal parts of the axons begin to degenerate, and that leads to these severe chronic pain conditions, like chemotherapy-induced peripheral neuropathy, CIPN, and diabetic peripheral neuropathy.
Yeah.
And for decades, the dogma was, well, the neuron had to generate and maintain every single one of its own mitochondria.
But that's the beautiful twist in this research. They aren't self-sufficient. They have an expert pit crew.
They do. Our source material confirms there are these non-neuronal cells called satellite glial cells, or SGCs, which tightly surround the DRG neuron bodies.
Okay.
And these SGCs are actively transferring healthy mitochondria to the stressed neurons. So the question for researchers became, how? How does one cell physically hand over an entire large organelle to another?
And this involves these specialized microscopic conduits. We're talking about structures that honestly sound like something an engineer designed, tunneling nanotubes.
Exactly. And these tubes are regulated by a protein called myosin 10, or MYO10, and they physically push these protective mitochondria into the struggling neuron. I mean, this completely redefines nerve survival.
Let's start with the architecture then. The DRG neuron. What makes it so unique?
Well, it's what we call pseudoanipolar. The cell body sits protected in the DRG, but its axon splits. One branch goes to the spinal cord and the other goes out peripherally, maybe all the way down to your toe.
And that distant terminal is where the damage often starts.
Precisely. So if that neuron needs a power boost way out at the terminal, think about it. Generating a new mitochondrion back in the cell body and shipping it down a 100-centimeter highway.
It's just incredibly slow and inefficient.
Right. So the SGCs offer this immediate local energy boost right where the cell body needs to coordinate its entire response. They physically envelop these neurons like a protective glove.
And this idea of cells sharing entire organelles, it really challenges that textbook concept that cells are, metabolically isolated.
It does.
So how did the researchers first prove this radical idea? How did they see mitochondria jumping ship?
They started in vitro, so in a dish, co-culturing mouse SGCs and neurons. They tagged the SGC mitochondria with a fluorescent dye called MitoTracker.
Okay, so they make them glow.
And they made them glow.
Yeah.
So if that dye showed up inside the neighboring neuron,
yeah,
well, transfer confirmed. It was immediate visual proof.
And what did the actual structures look like, the nanotubes?
They were stunning. Confocal microscopy picked up these tunneling nanotubes, or TNTs, connecting the cells. Some of them stretched over 30 micrometers long.
Wow.
And crucially, you could visually see the mitochondria, the little stained powerhouses, moving inside these tubes.
Okay, so here's where my inner skeptic kicks in a little bit. The data showed that something like 83% of the neurons received mitochondria?
That's right, 83.3%.
But only about 31% of them had a visible TNT connection when they looked. If the tubes are the highway, why is there such a massive discrepancy?
That's a really critical observation.
Yeah.
And the answer is that the TNTs are incredibly transient. They're temporary. Live imaging shows they form quickly, they do the transfer, and then they disassemble.
In minutes.
Often within just minutes. So while the transfer itself is highly efficient, the physical connection is like a temporary pipeline. It's here one moment, gone the next.
Which explains why you wouldn't see them all in a single snapshot.
Exactly.
So the vehicle is temporary. But is the process passive, or does the neuron have to sort of ask for the delivery?
Oh, the neuron absolutely signals its demand. The whole thing is activity dependent. When researchers used tetrodotoxin TTX to block the neuron's sodium channel.
Which basically silences it, stops it from firing.
Right. When they did that, the rate of mitochondrial transfer just plummeted.
But did it stop the tubes from forming?
That's the key part. It did not stop the SGCs from forming the TNTs, but it stopped the cargo delivery. This strongly suggests that the electrical signaling from the neuron is the distress signal. It's the call for help that triggers the SGC to transfer its power source.
Okay, so to recap this first part, we have these incredibly long sensory neurons in the DRG with huge energy demands.
Right, and they're vulnerable in diseases like DPN, CIPN.
And the satellite glial cells that surround them act as mitochondrial donors like a support crew.
And they use these tunneling nanotubes, or TNTs, as the vehicle for this transfer.
A vehicle that's dynamic, transient, and crucially, it depends on the neuron's own activity. It has to ask for help.
You've got it. That's the core mechanism.
So now the question becomes, does this whole system actually exist in the complex, messy, living anatomy of a person?
To get there, researchers had to be really rigorous and rule out other potential transport methods. So back in the Petri dish, they dissected 3 possible pathways using pharmacological blockers.
Okay, we don't need a heavy list of chemical names, but what were the 3 pathways, and what did the blockers tell us?
So the 3 suspects were, first, the TNTs, which we've talked about. Second, endocytosis, which is where the neuron basically engulfs the particle.
It just eats it.
Kind of, yeah. And third, gap junctions, which are these tiny protein channels that let small molecules pass between cells.
Okay.
And they found that when they blocked any of those 3, using things like cytokalasin B to mess with the TNT structure or pitstop 2 to stop endocytosis, they all partially suppressed the transfer.
So it's not just one thing.
No, it told them it was a coordinated effort. The tubes, the engulfment, and even the tiny junctions, they all contribute, at least in vitro.
But did the tubes themselves, the big structures, did they actually exist outside the dish?
Yes. They use some really high-resolution imaging scanning and transmission electron microscopy, or SIM and TEM, on both mouse and human DRG tissue.
And they found them.
They found TNT-like structures connecting adjacent SGCs and neurons. And the TEM images, they provided the key evidence. These smooth, tubular connections that had large, vesicle-like bulges inside them.
Bulges that were consistent in size with mitochondria?
Exactly. This confirmed that the structure is a real feature of human anatomy.
Okay, so the delivery truck exists in the body, but the final, the gold standard proof is showing the delivery happening live inside a living animal.
For that, they use this really clever genetic tagging in mice called MitoTag. What they do is they genetically label only the SGCs, the donor cells, to have a green fluorescent protein on their mitochondria.
So if a neuron starts glowing green...
You know exactly where it came from.
No other explanation. And what was the real-time proof?
Over time, the green signal just showed up dramatically inside the neurons. At day 5, only about 3% of neurons were green. But by day 10, nearly a quarter of them, 23%, had visibly received their emergency energy packages.
A quarter of the neurons. Wow.
And what's more, when they gave those same blockers, Cyto-B and pitstop 2 in vivo, they partially decreased the transfer, which confirms that both TNTs and endocytosis are active mechanisms in the living animal.
Does this protective mechanism ramp up when the nerve is actively distressed, like after an injury?
Absolutely. When they used a spared nerve injury model, which is a common way to induce neuropathic pain, the percentage of labeled neurons skyrocketed on the injured side of the DRG.
So it's demand-driven.
Completely demand-driven. And to really hammer home that activity link, they used a local anesthetic, bupivacaine, during the injury. And when that drug blocked the neuronal hyperactivity, the injury-induced increase in transfer was completely abolished.
Activity is the trigger, and injury is the amplifier.
That's a perfect way to put it.
So is this a specific, localized emergency response system, or does it happen elsewhere in the nervous system?
It appears to be unique to the peripheral nervous system. The sources confirm the transfer is predominantly unidirectional, from SGCs to neurons.
So they checked the central nervous system.
They did. They labeled spinal astrocytes, which are the central equivalent of these glia, and found minimal to no transfer to spinal cord neurons. This really makes SGCs crucial, specialized support structures for these long, vulnerable peripheral axons.
So to recap here, microscopic proof from SEM and TEM confirmed these TNT-like structures in both mouse and human tissue.
Right. And the MitoTag mice confirmed it in vivo, showing significant transfer over time, especially after injury.
An injury-driven transfer that depends on neuronal activity.
And it seems to be a unique peripheral glial mechanism, not something we see in the central nervous system.
Okay. We've identified the vehicle, the cargo, the signal, and the location. Now we need the architect, the molecular force that's actually building these tubes.
And that architect is a protein called MYO10 or myosin 10.
MYO10?
Yeah. Single nucleus RNA sequencing confirmed that MYO10 is highly enriched, specifically in these satellite glial cells. This protein is essential for forming the scaffolding of the tunneling nanotubes.
So if you target MYO10, does the whole protective mechanism just collapse?
It does. When they silence the MYO10 gene in SGCs, it drastically reduced TNT formation and completely blocked mitochondrial transfer in their co-cultures.
And what did that do in the whole animal?
Well, mice that had a partial genetic loss of MYO10, the myo10 plus minus mice, they showed increased pain hypersensitivity, which means this protective transfer is essential to prevent chronic pain states.
Let's connect this breakdown to those severe conditions we started with, CIPN and DPN. Where is the system failing in these human diseases?
It fails structurally and it fails chemically. In the chemotherapy model using paclitaxel, two things happened at once.
Okay.
First, the SGCs themselves suffered mitochondrial dysfunction. They started showing these shorter, stressed-out powerhouses. And second, the chemo caused significant structural separation. These enlarged gaps started forming between the SGCs and the neurons.
So the pit crew is damaged, and they're also physically pulling away from the athlete that needs help.
Exactly. And what about the human diabetes link? The findings were parallel and, frankly, devastating.
What did they see?
Analyzing human DRG tissue from diabetic donors showed that the expression of that key protein, MYO10, was significantly downregulated in their SGCs.
So the architect is gone.
The architect is leaving the building. And structurally, the gaps between the neurons and SGCs in diabetic patients were massive, up to 1 to 4 micrometers. Compare that to the healthy, tight 0.1 micrometer connection.
That's a huge difference.
It's a chasm.
Mm-hmm.
And this disconnection leads directly to reduced mitochondrial transfer. The lifeline is broken.
Okay, here's where it gets really interesting for me, because this creates a major paradox around small fiber neuropathy, or SFN.
Right.
SFN is the common degeneration of the small, painful nerve fibers in diseases like DPN and CIPN. So why are the small fibers the first to die if this whole system is designed to save them?
That is the central and very counterintuitive finding. The research confirmed that after an injury, the neuronal distress signal, the hyperactivity, comes primarily from the small-sized neurons, the pain-sensing nociceptors.
Logically, they should be first in line for the rescue package.
You'd think so,
Yeah.
but they're not.
They're not.
They are not. The SGC-derived mitochondrial transfer preferentially protected the medium to large-sized neurons. It's a size-selective rescue operation.
Wait, so the fibers that are literally screaming the loudest for help, the ones that are actively injured and sending the highest distress signals, are the least likely to receive the energy package.
That seems so.
So SFN pathology, the death of these small fibers, isn't just a general energy failure, it's a failure of the delivery system's specificity.
Exactly. This discovery provides a mechanical explanation for SFN, this critical SGC protective mechanism. For whatever reason, maybe structural constraints, maybe something about MYO10 distribution, it bypasses the very small nociceptors that need the mitochondria most. And it leaves them vulnerable to degeneration. It completely reframes how we think about pain fiber loss.
Okay, so if the pathology is a broken delivery system, reduced MYO10, structural separation, this preferential targeting, can we fix this? Can we boost the delivery or just force the issue?
They tested 2 really powerful strategies. The first was called adoptive SGC transfer.
What does that involve?
They injected healthy human SGCs directly into the DRG of diabetic mice. And this not only alleviated the mechanical hypersensitivity, reduced their pain, but it proved the effect was MYO10 dependent.
How so?
If they silenced MYO10 in those healthy SGCs before injecting them, the pain relief vanished.
So the transfer mechanism itself is the engine of recovery.
It absolutely is.
What about bypassing the cell infrastructure entirely? Can you just give the struggling neuron the power source directly?
That was the second, and I think even more exciting, strategy: direct mitochondrial transfer. They isolated healthy mitochondria from SGCs and injected them directly into the DRG of neuropathic mice.
Both the chemo and the diabetic models.
Both models. And the results were highly encouraging. The healthy mitochondria mitigated the mechanical pain, and this is a key marker. It improved the intraepidermal nerve fiber density, the IENF.
Which is a measure of nerve regeneration in the skin?
It's a direct measure of nerve health and regrowth.
So did these isolated mitochondria still need the TNTs to get into the neuron?
No, and this is a key technical insight. They found that this direct uptake was mediated entirely by endocytosis, The neuron just sort of engulfed the free-floating powerhouses.
So the TNT blocker had no effect.
Correct. The endocytosis blocker, pitstop 2, stopped the uptake, but the TNT blocker, cyto B, did nothing. This means if you can deliver the cargo directly, you can bypass the need for the SGC's whole MYO10-driven infrastructure.
And I'm guessing the quality of the power source is paramount.
Absolutely. When they used mitochondria isolated from dysfunctional diabetic SGCs or mitochondria they had treated with toxins, there was no protection. The therapeutic potential relies entirely on delivering a healthy, functional mitochondrion.
So what does this entire deep dive mean for the big picture of chronic pain?
Well, this research uncovers a fundamental and previously unrecognized role for peripheral glia. They are the dedicated energy support system for these long, vulnerable sensory neurons.
They're transferring their powerhouses via this MYO10 regulated highway.
Right. And diseases like DPN and CIPN disrupt this support by damaging the SGCs and causing them to physically detach, which leads to neuronal degeneration. The crucial takeaway is that the cell's powerhouse is a shared, networked resource, and we now have a mechanical explanation for why small pain fibers die first.
And that leads to the final provocative thought for you to chew on. If we know this natural survival mechanism preferentially fails the small, vulnerable pain fibers, leading directly to small fiber neuropathy, then the next generation of targeted treatments has to fix that bias. We've just seen that direct mitochondrial delivery works via endocytosis, completely bypassing the flawed TNT system. So could future therapies use nanoparticles or maybe modified viral vectors that are engineered to specifically target the surface markers on those small nociceptors? Could we finally deliver those healthy energy packages precisely where the cell is screaming for help? I mean, it creates a complete roadmap for ending one of the most intractable chronic pain conditions we face.